Welcome back. Before the break, we've been looking at gene therapy. That involved taking a healthy, functioning, normal gene and inserting it into a cell where there was a faulty gene. Now, another way of changing the genetics of a cell or using healthy genes to combat the situation where there might be non-working or dysfunctional genes. Another way of doing this is by genetic engineering. So let's have a look at what genetic engineering is and the role that bacteria play in genetic engineering. So, what is genetic engineering? Well, it involves removing a gene from one organism. That organism is called the donor. So we're going to take a gene out of the donor and we're going to transfer it into a recipient organism so that the recipient organism now has this gene in it that it didn't have originally. It now has a gene from some other organism, and we call this new organism a transgenic organism or a genetically modified organism. Sometimes you might see that referred to as a GM organism, genetically modified. So, simple. We're going to take a gene from one organism, we're going to remove it, we're going to put it into the gene of a recipient organism so that the recipient now we call genetically modified. Now, this idea of genetically modifying an organism might result from for two reasons. It could be that we want to be able to give this recipient organism some advantage that the donor organism originally had. So let's call the donor organism A. And let's say A is a particular kind of flower, a plant that has little flowers that function extremely well in icy regions. They grow in the snow, so they cope with frost and extreme cold very well. Let's imagine that the recipient organism, B, is a strawberry, for example, that doesn't cope very well with frost. And strawberries will die in very, very frosty areas. What wonderful advantage could we confer on the strawberry plant if we could take the gene that resists frost and put it inside the strawberry? Then we could grow strawberries in very cold conditions, and frost wouldn't harm or wouldn't destroy the strawberry plant. And that is what one application of genetic engineering. We are giving the recipient organism an advantage that the donor organism originally had. And as I said previously, then the recipient organism is then called the genetically modified organism. Genes are removed from B and inserted into A using microorganisms. And these microorganisms usually are the bacteria that we spoke about earlier. Now, a second reason for performing genetic engineering or for doing genetic engineering is that sometimes we require very large volumes of a particular protein to be made. Scientists can actually use bacteria to produce large volumes of protein for our use. For example, insulin, growth hormone, and various other medications. Some of you may have had different vaccinations for things like hepatitis, and these vaccinations are protein-based, and they are made through the process of genetic engineering. What genetic engineering involves in this case is inserting a length of DNA from one organism into the microorganism, usually the bacterium, and then that bacterium produces this foreign substance. It's foreign to the bacterium, but it's needed by humans, like, for example, the insulin, the growth hormone, the vaccination. and that those bacteria just go ahead and produce this new substance. The substance then can be purified and it can be used to our advantage. Now we're going to look at the situation involving diabetes, type 1 diabetes, but the same situation applies for growth hormone or the manufacture of vaccinations. It's the same process. What we have in a type 1 diabetic is the cell requires glucose to get into the cell from the blood in order to produce ATP. Now, the problem with type 1 diabetes is the glucose can't get across the cell membrane. And the reason for this is in normal people who do not have diabetes, insulin acts as a key to allow this glucose to move across the cell membrane. If you are a diabetic, a type 1 diabetic, you do not have insulin, and so you cannot allow, or not you cannot, it won't allow the glucose to move from the blood into the cell. And so what happens is, therefore, you can't make ATP. And in order to get that energy, your cell starts to break itself down. So originally, diabetics injected insulin that was made from ground up calf or pig pancreas, and there were often complications. For example, the person could be allergic to the pig insulin. They could have problems with religion, having the pig insulin inside them, and it's an expensive process as well. And so a need arose to produce human insulin via the process of genetic engineering. And when we come back after the break, we're going to go through the process step by step. You might want to get yourself a piece of paper so that you can copy down the various steps associated with genetic engineering of insulin.